Okay, so back to our questions. We've spoken a little bit about what DNA is made out of and what kind of structure it has, and where it is. It's in the nucleus of a cell. Now, the next question: how does it fit? How does it fit in a cell? A cell which essentially, is so much smaller than the length of the DNA. And this is really, there were two mind-blowing aspects of the discovery of DNA. And the first one, the most fundamental one, was that DNA is the same in every living thing. Not the sequence, but it's coded for in the same letters, A's, C's, T's, and G's. And what that really means is the code of life, the thing that makes us who we are, that makes a plant and a fish, a human a human, the thing that makes us who we are and gives us our characteristics is written in the same language. The code of life is written in the same language in all living things. And what I mean by that is every living thing has this code, and it's written in A's, C's, T's, and G's. The adenine, cytosine, guanine, thymine. The same chemical in that sequence of those. It's just the sequence, the order of them that is different. And that had, just such fundamental impact on biology because it suggested a few things. Number one, it suggested that we probably all came from a common ancestor. It was really a great support of that idea that we all evolved from a common ancestor. Number two, it just meant that, the implications of what we could do with this are just, mind-blowing. It's, in theory, we can take a sequence out of one living thing that gives it a characteristic and put it in another living thing. So, for instance, a jellyfish, we could take a sequence out of a jellyfish that makes it glow in the dark. And we could take that sequence and put it in a rabbit, and in theory, the rabbit would glow in the dark. And that might sound ridiculous to you, but that's actually an example that's already been done. We've taken a gene, a bit of DNA, out of a jellyfish, and that bit of DNA actually codes for or makes that jellyfish glow in the dark, and we've put that into a rabbit. So in theory, the fact that every living thing, that its code of life, its DNA is written in the same language, those A's, C's, T's, and G's, means, it just has such wide-reaching applications and potential for us to do things with that it is really probably one of the most fundamental discoveries in science ever, but definitely one of the most fundamental discoveries in the last 100 years. And it's such an exciting time to be in biology because of this. Because the DNA structure of DNA was only discovered or was proven by Watson and Crick. What was it in the late 50s? And it really means that since then, we're actually only now starting to discover and appreciate what we can actually do with that or use it for. So there's so many discoveries and so much being discovered at the moment, and there's still so much that is yet to be discovered in the field of genetics and molecular biology. So such an exciting time to be in biology. But the second fundamental mind-blowing thing was that this DNA must be an incredibly long molecule. Now, you may have learnt about starch when you did maybe the digestive system and how starch is a chain of sugars. And that's a chain of sugars of, several thousand maybe sugars long. But DNA is millions of these base pairs long. Millions. In fact, if it was just one long strand, it would be over 3 billion base pairs long. So how do you fit 3 billion base pairs, a piece of DNA that's 3 billion base pairs long, into a cell? A cell's not nearly big enough. In fact, if you took the DNA that's in one cell and just stretched it out in a straight line, it would be between 1 meter and 2 meters long. Now, none of the cells in our body are that big. Maybe some of our nerve cells are 1 meter long, but most of our cells are much, much smaller than that. So how do we fit the DNA into such a small size? And the DNA is actually found in the nucleus, which is even smaller again, smaller than a cell. And the answer is it has to somehow be packaged up. And we're going to look at that in a second. But that DNA really needs to be much shorter. And the first way we do it is, for a start, in humans, we don't just have one long length of DNA. It's actually been split up into 46 pieces. So that's going to help also fit it into a smaller space. Our DNA has been split into 46 pieces. Now each of those pieces basically condenses, it coils up. So a piece of DNA, which looks, like our double spiral, an incredibly long length, alright, incredibly long spiral here, wraps itself around a couple of proteins, alright, and those basically coil up again. So it coils up, this whole long strand of DNA coils up tightly into basically a sausage-like structure. So if we draw a sausage-like structure, I'll use this one, so that those long lengths of DNA coil up into essentially what looks like a sausage-like structure. So this is basically a DNA sausage. Now this DNA sausage has a special name. It's called a chromosome. So when you see the word chromosome, I literally just want you to think DNA sausage. So it's this thing here is a sausage made up of a massive long length of DNA coiled up. Now we see these best, these chromosomes. So the DNA isn't just randomly floating around in a cell like a bowl full of spaghetti. It's highly organized. And especially when the cell has to divide, the DNA then has to be extra highly organized. And the reason for that is when we make copies of cells, and when we have to copy the DNA, we're going to have more DNA for a start, so we need to be organized about that. But we're also going to have to separate the DNA out when the cell divides, so that each of the new cells has a copy. And because we have to separate the DNA, and we want to make sure that each cell gets a complete copy, we need to make sure they don't get all tangled up and things like that. And so during cell division, when a cell grows and divides and it has to copy its DNA, the DNA becomes extra organized. And that's the best time we can see these DNA sausages. But because we've copied the DNA, we don't have a single sausage, we actually have a double sausage structure. So it looks like two sausages stuck together. So two sausages stuck together, the double copy here. This is often what we actually call a chromosome, because this is when we actually see them best under the microscope. So a chromosome is generally used to refer to this double sausage structure, and technically one sausage is actually called a chromatid. I wouldn't worry too much about that, but a chromosome is a DNA sausage, so coiled up, a structure that's made out of coiled up DNA. If we took that and unwound it, we'd have a massive long length of DNA. And we see them best during cell division when there's a double copy. So it's this double DNA sausage, each sausage containing an exact copy of the other, that we call a chromosome. Now the bit in the middle where they're attached is called the centromere. So there's a little bit of protein here holding the two sausages together. But at some point the sausages will separate when the cell divides, and each of the resulting cells will just get the one copy. And then it will actually uncoil a little bit, the sausage relaxes a little bit, and so you don't see them so clearly anymore under the microscope. So guys, where is DNA? In the nucleus, and how does it fit? Well, essentially it's coiled up, it's packaged up, and especially during cell division we can see it packaged up into these double sausage structures where there's the two copies there coiled up tightly into a sausage. These double sausage structures we call chromosomes. So anyway, let's have a look at another couple of little videos, and we'll come back and we'll ask this question: what does DNA actually do? But we'll do that in the next tutorial.